Welcome back, everybody. So, as promised, we reached 250 subscribers. I can't believe it. We're going to go through and we're going to pick apart my dissertation. We're going to talk about how disgusting it looks and the things that we could do better that we've learned over the years. So, let's get into this. I think enough of you have found my name on the internet, so I'm really not going to black that out. Anything that I will black out is just towards the end, which is acknowledgments about my son and my wife, because I think they're entitled to their own privacy and we don't need to mention them. But everything else, this is my name, Justin Darcy. I worked in the lab of Andre Barkey. You could look up Andre, you could look up me on the internet. That's fine. This is the culmination of my doctoral work. And if you kind of want to know the caliber or some of the caliber needed to get into Harvard, I think that there's a lot of different people with different qualities that get in. I published 3 different first author papers. This presentation is on 2 of them. One of them I didn't quite talk about because it was unrelated to this. I had several other review articles that I published, and I also had a whole plethora of co-authorships as well. But, without further ado, let's actually get into this presentation and talk about my dissertation and how my defense presentation could have been a lot better. So the title of my presentation was Energy Metabolism and Aging. I have my name, advisor, dissertation, and I defended mine on May 25th, 2017. The first thing that I'll say about this is the colors for this presentation were horrendous. I don't know really what I was thinking at the time. I thought that I was being kind of edgy, but really the font, we should stick with a plain font like Arial or Times New Roman, and we should be using more of like a white background, maybe some accent colors. But these kinds of colors like the grays and these dark blues, they're kind of hard to see and the writing isn't very clear. So I wish that I had changed that. So obviously my thing is about aging. So I had a slide in here, what is aging and why I study it. This is totally not necessary for a slide. I don't quite understand why I put it in here. And there's much better transitions that I could have used, especially if I was just verbally doing it. But you know what, I had it in here and I kind of talk about the different components between genetics and environment that play a role in aging and how it can lead to different, age-related diseases. Okay, just an overview of aging. This is more just an overview of aging and kind of what the goal of, the biology of aging is. I think that all of this is okay, but I think that overall what I would actually recommend is that a lot of these were homemade kind of slides that I had put together or modified from slides that other people have had. And what I actually think is a better approach is to actually go into publications and copy and paste over, take a screenshot of the actual figures that they have and just cite them. And I kind of stayed away from that in grad school. I don't know. I felt kind of like I was cheating by using other people's graphics and that it really wasn't mine and that I should put in the effort to try to make some of my own. And really what I kind of figured out as I was going through later in my career is that the graphics that I make, at least, some of you may be experts, but a lot of the graphics just are not nearly as good as what are in the journal articles. And so your best bet is to honestly go through and copy those and put them into your presentation and don't waste the time making your own. I mean, unless you're super, talented in Illustrator, it's just not worth it. So I study these cute little mice, these long-lived mice. If you're curious about what an Ames dwarf mouse is, you can Google that, but there are these really cute little dwarf mice that live really long. And I have some background about them. And this is, again, back to what I was saying. This is a schematic that totally could have been copy and pasted from 1,000 different articles. And I decided to make one myself. And it's the most convoluted, roundabout presentation that I've put together. And this could have been so much clearer. It really could have. I could say that I did a good job with the red and green, but even with that, nowadays, I think people try to stay away from using red and green because there's colorblind people. So there should have been a different color scheme that was designed. It wasn't the best. So here I actually did pull from Wikipedia, which is honestly not the best place to pull from because there's, again, this is about thermogenesis. There's a bazillion papers that have illustrations on this, and I'm not sure why I pulled from Wikipedia. I think it just was a newer area that our lab didn't focus on, and so I tried to pull that in. But I think that if you're gonna pull any of these in, first of all, what you should do is you should make it like the whole slide, not just like part of it. You could even add like a black border around the edge of the picture to kind of like embolden it. And I think the captions should have been better. This was just, again, not very well put together. And then I just go into the data, I guess. I don't know what I was thinking. Like, I didn't even give an explanation of like what the study design is or like what the rationale is. Like, this is terrible. Like, there should be at least, from here we talk about, why these mice are long lived and that maybe energy metabolism plays a role. And so I introduce thermogenesis and then I just go into the study, but there's no connection. There's no like how I'm going to study this or like what I'm hoping to get out of this. Like, I just jump right into the data. Like, who made this? I mean, I guess I did, but this is just not very good. So we go through the data. Now, I will say one of the things about the data that I did well was all the figures are easy to read here. So you could be in the audience and you could see, what the legends are. You can see, what the p-values stand for. You could see where the citation comes from. The title of the slide is a statement. It's not just some kind of broad thing. It says, Amesdorf BAT thermogenic mRNAs increase. Like, it's giving you a definitive statement of what this slide is supposed to be showing. That's good. The color scheme is good. It's easy to follow. The stars that are above the bars are easy to see, so it's easy to understand what's actually happening and what the statistical significance is. These are all good things. And I know that when I went through this, because I practiced it a bunch of times, I did take my time and actually walk through each specific, section of genes that I was looking at. So there was actual definitive, clarity in what I was saying. So that part is good. We go through some H&E stains. There's nothing wrong here. Okay, so here, in this part, I actually do give like an overview of the scientific study of what I'm doing. Why I didn't before, I don't know. But this is good. There could be a better design for, what we're doing in the study, but this, it gets the point across. This is what we're going to be doing. We're going to take these mice and we're going to do this kind of surgery and we're going to wait for them to recover, and then we're going to examine the different effects of the metabolism. Okay, this slide is okay. There is, good color coordination. The biggest issue with this slide is that it was pretty much lifted from the publication that I put it in. And the reason that this is an issue is that I have these A, B, and C's listed on here. And the issue with this is that this is because this was done with a two-way ANOVA. And what it's basically saying is that anything with an A is different than anything with a B. And anything with a B is different than anything with an A or a C. So it's kind of giving the differences between the groups. But I don't have any kind of figure legend anywhere put out that basically shows what that means. And so because of that, I think people got confused about what are these A, B's, and C's. And why are there no like stars? And I remember that when I went to go do my postdoc interview, in one of the labs, I had given this similar presentation to this, and I had this kind of similar ABC schematic. And I remembered like the PI that I was interviewing with was really confused. Like, what does that even mean? And I tried to explain it and they sort of got it, but it just, I should have made this clear. The other thing is like, what is with this y-axis? Goes 0 to 30 and then it's like skips and then it goes to 30. Like, what? Like, why didn't I just make it from 0 to 40? Okay, I'm not quite sure why I made it like that. These are a little bit easier to follow, but again, these A, B's, and C's are kind of confusing, and these y-axes are really hard to follow. Again, here I actually lay out what the experimental design is because this isn't an experiment that many people are used to, but I don't know why I didn't do this from the beginning. All right, so then we talk about mechanism here, and I'm basically explaining what I think is the mechanism of why their energy metabolism is different and kind of like how I'm going to interrogate this. This is actually really good. When you're doing your defense, or even when you're applying for a postdoc, this is actually something that's important, is being able to specify, now that I've completed a project and I have a set of data that is now a story, that I have something that I'm going to build off of. And what is the mechanism that I think is driving this? If I didn't already answer it in the first part, now how do I further interrogate this mechanism? And so this is actually saying that, this mechanism isn't just something that I just dreamed up, that I actually have a hypothesis and a plan to test it. And that's what this back half of this is. So, okay, again, we go back to the A, B, C's, and D's. So the G's here represent in the two-way ANOVA that there was an effect on genotype of the mouse. So whether they were like the control or the knockout mouse. The T represents that there was an effect by temperature. So these mice are housed at different temperatures. And G by T shows that there's an effect on genotype and temperature, that the genotypes respond differently to temperature. So you could see like the white bar is going from low to high to the blue, and then in the stripe bars it's the opposite. But anyways, this is confusing as all heck. Like, nobody in their right mind that's following this is going to have any idea what this means. You're just not. And there wasn't any kind of legend put into this, so that could have been a lot clearer. Again, these G's and T's, I don't know where this came from. And then I jump kind of into the conclusions, and I kind of talk about the conclusions a little bit, but really what I miss in here is talking big picture. These are like bullet points of like, these are the three main findings. But what I should have included, and I got a lot better at including as I grew and matured, is some sort of a visual aid that kind of walks together and puts the whole picture together. And that's really what I'm missing here in the conclusion, is that I'm sort of just talking bullet points of this changed and that changed, but I'm not actually going through and giving big picture like cohesiveness of these things changed, which caused this to happen, and then this is the overall effect. And that kind of cohesiveness is something that a lot of grad students desperately miss. And I myself also missed it. And I think that as you progress through your postdoc and further in training, you'll get better at it. But if you can, by the time you're at the end of grad school, be able to bring that cohesiveness together and show one illustration that kind of wraps everything together in a neat bow, you're doing pretty well. And then the acknowledgement slide, which is just the acknowledgement slide. Nothing special here. But yeah, I would say that that's pretty much going to wrap up exactly my dissertation. So if you ever wanted to look and see what exactly does my dissertation look like, well, now you have inside access to it. So with that, I'll catch you guys in the next one. Bye.